To download Protospacer, go to protospacer.com. Click on the download link and then click on the download software button. It'll take you to SourceForge, where you can download the actual archive file. I'm going to create a temporary folder for this exercise, where I'll store all of Protospacer files. Next, I'm going to go download a database to use with Protospacer. I want to download the human genome, but it's slightly different from most of the other databases. It's too large for SourceForge to host, so it actually has two parts to it, and a slightly different installation process. These instructions are available on SourceForge in the README file. I'm going to need to download main.zip and index.zip. Once this process is complete, I'll simply copy it into the temporary folder that I created on my desktop. Now that that's done, I'll unpack the Protospacer software. To install the database, I need to copy main.zip into the data directory of Protospacer Workbench, and double-click on it to unpack it. Since it's a two-part database, I need to copy index.zip or move it into the base data directory, and once again unpack it. The database is now installed and I can run Protospacer Workbench. To do this, I need to right-click on it and click Open. After opening Protospacer, it takes me to the main screen and shows me my available databases. Since I've only installed Homo sapiens database, that's the only one that's available. You can click on it and then click Load to load the database. Once this is done, you're taken to the main screen. Now I've gone to the Information tab, which shows you the name of the database, the dates it was created on, the size, some notes or further information, the reference names, and identifiers for genes within the database. Defining a region you would like to target is the first step in guide-RNA design. The second step is filtering targets within the regions, then selecting further targets you would like to analyze and analyzing them. But to start, to add a target region, you double-click on the row. You can add a genomic range, specify chromosome, start, stop, and strand you'd like to analyze, or a distance from a point, a chromosome, a coordinate, and the distance from the coordinate you'd like to analyze within, or a gene identifier. Note that the GUN gene is not accepted, but the ensemble gene identifier is. We could add a gene by its attribute. If we specify gene ID, again, GEN doesn't work, but the ensemble gene identifier does, and is auto-completed. However, the gene name, the ensemble gene ID isn't recognized, however GEN is. We can also add a region by sequence similarity. The sequence you add needs to be between 20 and 1000 nucleotides in length. If anything is incorrect in the sequence, the bar will turn red, and you will not be able to map the sequence. Let's add the GEN gene. And analyze both its strands. You'll see here information for the target region. There are 534 targets. Note that the region has children. It has genes, exons, start and stop codons. Let's say we want to target both the start and the stop codon. We can click on the Children button, select them, and click Include. Since we only want to analyze the start and the stop codon, we can remove the GEN gene. I want to analyze 100 nucleotides up and downstream of each codon. So to do that, I edit the table directly. Notice at the bottom, the text in blue is changing dynamically as we change the target regions. There are 54 targets in this target region. As you can see in the top right, I have 54 unfiltered targets within the regions of interest I specified previously. In step 1, I can filter by nucleotide content, and in step 2, I can filter by the Wunsch root activity score. In step 3, I can specify that I want all 20 nucleotides of the guide-RNA to be unique. In this instance, 16 targets passed. The results screen shows all passed targets. Here I can see targets for the stop codon and the start codons that I specified. I'm going to select all and click on Mark, then proceed to further analyze these 16 candidates. This is the Mark Targets analysis screen, where you get to manipulate and analyze your data. It's probably where you'll spend most of your time. To begin, since I have two regions, I'll color code them using the target analysis functions to the bottom right. I then wish to analyze them. I'll use Bowtie to do this, to analyze for off targets up to 3 mismatches away. Because of Bowtie's limitations, 3 mismatches are the maximum I can use. It takes 6.25 seconds to do this. Now, for every target, I have results for the guide-RNA design itself, for the on-target and off-target statistics. After double-clicking on a target, I've been taken to the results screen for the guide-RNA. There are many statistics shown on the screen. Take your time, pause the video, and read the descriptions. This plot provides a graphical summary of the guide-RNA's performance. The title of the plot is the intended target site. The x-axis is the rank of each target site, with the leftmost targets being the most likely. The y-axis is a 0 to 1 score whose interpretation changes depending on the data. The pink line is percent identity of guide-RNA to target. The yellow data is the Wunsch root activity score, and the green is the pairwise score. The blue triangle always specifies the rank of the on-target site. This plot represents a decent design, with rapidly decreasing scores from the on-target. We will see an example of a poor design later on. Looking at each guide-RNA report to determine the best guide-RNA design is certainly the best option. However, it can be tedious to do with large lists. Protospacer provides the ability to aggregate and rank each guide-RNA design. In this screen, I am going to select the top 6 designs. Now that I have 6 good candidates, I want to write a note on them. You can put any descriptive data that you'd like into a note. It's especially important if you want to share your saved list with a colleague. The colleague will need to read the notes and understand why the targets are important. Your notes will persist throughout the session, but if you close Protospacer, you will lose them. To save the target list, the color codings, as well as the notes, you need to save the targets to a file. Having saved the targets, I close Protospacer and open it again as a demonstration. To load a target list, you need to load the database it came from. You can then go directly to the Mark tab, click on Admin Options, and load targets. If you view the notes, they are still there. I now want to have a look at the plots for each target. To do this, I'll first open the targets by double-clicking on them. I can quickly navigate between the tabs by using the arrow keys on the keyboard. Notice that guide-RNA 3 only has one target in it. This is because we use bowtie to do the analysis, which only has 3 mismatches maximum. If we want to find off-targets that have more than 3 mismatches, we need to use one of the other algorithms. Here I use Razor S3. Razor S3 takes some time. Here, 463 seconds. Now that the analysis is complete, we can see that Razor has supplanted the bowtie results. And if we double-click on one of the targets, we see the report. For the guide-RNA, the plot has been updated as well, with a lot more off-targets. We can open the plot in the browser for detailed analysis. The plot is interactive. I'll now close all of the plots and demonstrate that even if we remove the results, if we load the target list again, the Razor S3 results will still be present. I now want to select the top 2 and analyze them further. These will be the candidates I take to the bench. If we look at the plots, we can see they're pretty good. But what does a bad plot look like? To do this, we'll add a custom guide-RNA. A guide-RNA that we don't know whether or not it's present in the actual genome. The guide-RNA needs to be 20 nucleotides in length and won't be accepted otherwise. I analyze the guide RNA with Razor S3 again. And again, it takes some time. If we open the custom guide RNA, we can see that there are 147 targets, exactly matching it. And plotting it shows what a poor guide RNA design looks like. Compare it to the other guide RNAs. I'll append back the list that I removed. When appending a file, the color coding and notes are not appended with it. So if we want to get the color coding back, we need to redo it. I'll save this file over the last one. Protospacer's databases are file-based and dynamically increase in size as you use Protospacer. So where are these files stored? If you want to share your results with a colleague, you need to know this. Go to the Data folder under Protospacer Workbench, then the database itself, and then Results. For each guide RNA design, you will see a folder. Here, you see the custom guide RNA design we just added, as well as several others that belong to the genome itself. We can compress these 17 results into an archive. This archive can be backed up for later use, or sent to a colleague for their own analysis, provided that they have the same database. Databases are the same, provided that they were created from the same FASTA sequence. If you go to the Information tab in Protospacer after loading a database, you will see that there's such a thing called a hash key. This is the MD5 checksum for the sequence data of the genome. Even if one nucleotide is different between genomes, this MD5 checksum will be different. This is for peace of mind and to ensure that results from different genome versions cannot be mixed. Finally, I want to show you how to export data from Protospacer. If you double-click on a target, it'll take you to the guide RNA design report. To export the likely targets for that guide RNA, click on the Export button to the bottom left. I'll export the targets for both of these candidates. The files that are created are tab delimited. We can open them in Excel, and they'll appear as a table. They are ranked top to bottom as the most likely targets. And the metadata on the top is our intended target. I've highlighted in yellow the predicted target and the intended target, and we can see that the top ranked predicted target is our intended target. I'll do this again for candidate 2. For candidate 2, I'll create a new tab in Excel and then open the file as a text file. Select all, copy it, and paste it into Excel. Again, the same structure applies. The target is written on top of the table, and all potential targets for the guide RNA are listed in the table, ranked from most likely to least likely. This concludes the guided tour through Protospacer. If you would like further help, please don't hesitate to go to protospacer.com. Thank you for your interest in Protospacer Workbench, and good luck.